Thank you very much for giving me this opportunity. Today, I would like to give my presentation titled Comparison of Cryptochrome Structures in Mammals Living in Extreme Environments. Cry4 in European robins is believed to act as a magnetic sensor, and some evidences have been provided until today. Similar reports have been made not only in birds, but in reptiles such as sea turtles, and insects such as monarch butterflies. Various animals, including birds, reptiles, fish, crustaceans, and insects, are believed to use magnetic fields for their navigation. On the other hand, although mammalian cryptochromes are known to have a function to control circadian rhythm, they are not thought to act as magnetic field sensors. However, it can be imagined that mammals in extreme environments such as in snowfields and in the deep ocean, need magnetic field sensors more than circadian rhythms controllers. Now we compare the cryptochrome structures of mammals in extreme environments, with the cryptochromes that are known to act as magnetic field sensors, in order to check if they have a magnetic field sensor-like structures. Second, we investigate the effect of these cryptochrome characteristic structures to the stability and other aspects of the three-dimensional structure of themselves. In the primary structure comparison, we compared snow ambient organisms. For snow environmental organisms, we compared Cry2 of polar bears, brown bears, black bears, pandas, and dogs, and found no structural differences in the Cry2 of these animals. Next, we compared the Cry2 of snow leopards, panthers, and domestic cats. The results showed that snow leopard Cry2 has a distinctive primary structure compared to domestic cat and leopard. We estimated 3D structure of snow leopard Cry2 and domestic cat to compare their 3D structure, and highlighted the characteristic structure of Cry2 of snow leopard. The left panel shows the 3D structure of the snow leopard Cry2, which has the characteristic structure, and on the right panel shows the 3D structure of domestic cat Cry2. These results suggest that the characteristic structure of snow leopard could be hung across the FAD binding site, where no particular structure is estimated for the Cry2 of domestic cat. In this slide, we show the results of the ISMB and cross-correlation analyses. The comparison between the results for Cry2 of snow leopard and domestic cat showed that the Cry2 of snow leopard showed a large variance of ISMB at the characteristic sequence and its N-terminus. On the other hand, a broad variance among C-terminus region of domestic cat was not inferred on Cry2 of snow leopard. Here we show the results of the principal component analysis for the 3D structure of snow leopard and domestic cat Cry2 and the cumulative contribution ratio. On the left is the principal component analysis plotted over time. By checking the sum of the proportion of each principal components, PC1 and 2 are enough to explain the major parts of the protein's behavior. Then we will discuss the results of projecting the first and second principal components of these principal component analyses. Here we have produced and compared the movements of the first and second principal components from the results of the principal component analysis. It was suggested that movements of features that were not seen in the first principal component were expressed in the second principal component in both of the results for a snow leopard and a domestic cat. And the movements were restricted on the N-terminal side of domestic cats, and on the alpha helix just before the specific sequence in snow leopards. Based on these results, we found that Cry2 in whales share more primary sequence features with Cry4 in fishes than Cry1, and that the structure of Cry2 in bears is conserved within species regardless of environment when examined. Furthermore, in the primary structure of snow leopard includes a characteristic sequences that is not present in domesticated cats, neither leopard. Although the existence of the characteristic sequence of Cry2 of snow leopard, it does not affect the overall protein structure. Neither the local molecular fluctuations suggest that the stability of the N-terminal side may have been altered. In our future, in order to analyze the effect of the unique structure of snow leopard on the function of cryptochrome, we will perform molecular dynamics simulations for FAD-bound Cry2, to estimate the effect of the FADH radical stability of the structure. We will also investigate the functional structures shared among Cry2 of whales and bears and Cry4 of fishes and investigate the effect of temperature on the structure. Here is the summary. We compared cryptochrome structures and predicted the similarities between Cry2 of cetaceans and Cry4 of fishes, and the discovered unique structure of snow leopards. Thank you very much for your kind attention.